Hi, this is a video for Tresham Year 1. This is your physiology unit for the digestive system. Today we're going to have a look at food groups tests. So the first one I'm going to do, because it requires a Bunsen, which I've got next to me here just out of your view, we are going to test for glucose. Okay, so I've got my beaker of hot water there. I've got a known glucose solution, and the reagent we need for this one is Benedict's. Okay, so it's a blue solution because it contains copper. Okay, so that gives it its blue color. And what we're going to do is we are going to get our known solution of glucose in a boiling tube. We're going to add a few drops of Benedict's. Okay, so I'll put the Benedict's next to the glucose. Okay, and then I'm going to put that into my warm water. Okay, and I'm just going to give it a warm through on the Bunsen. Okay, I've got it on an orange flame. I'm now turning it to blue. Okay, let's see if I can just pan for just a second. Okay, so I've got, you can't see the blue flame. Okay, I've got my solution in there. Okay, and I'll show you the result. We just let it warm through, and what should happen, because it contains the copper 2+ ion and therefore has the faint blue, it's a test for reducing sugars. So the sugars can change the state from copper 2+ to copper+, which will be red. So once it's started to warm through, it will go a dark red color. Okay, so while that's going, we're going to do the next one, which is a test for starch. So I'm just going to move the glucose across. Okay, and if you look here, we've got starch. Okay, now the one we need with starch is iodine. Okay, so we've got iodine here. Don't need heat for this one. So I've got my solution to test in here, which is a known starch solution. And again, the same as before, I'm going to add a couple of drops of my iodine. So that's a negative result at the minute. And you can see with the addition of the iodine, it goes to a lovely blue-black colour, which is a positive result for starch. So move that one back there. That's your iodine and starch. Okay. The next one we're going to do is for protein. So going through our food groups. So we've got a known solution of protein here, and I've put some into a test tube. Okay. Now this one is in two parts. So we need to add two solutions called Biuret 1 and, funnily enough, Biuret 2. Okay, so we're going to add the first one. Just need a couple of drops of each. Okay, and then with the addition of the second one, so we've got a negative test. And with the addition of the second one, I can see it. Not sure how clear it will be to you, but there is a definite lilac-y colour. Add a little bit more just so I can get it stronger so that you can see it. Yeah, there it is. Okay, can we see that a lot better? Yeah, look at that, lovely. Okay, so we've got a nice light lilac colour. I've got it in a picture here for you. Okay, so that would be our positive test for protein. Just as we've finished that one, I am going to turn my Bunsen off and show you the result here. Okay, just let it cool for a second. And then very carefully. So this is our Benedict's solution from before. So that's the copper blue goes to the kind of orange brown colour we've got here. So that is a positive test for glucose there. Okay, so the Biuret 1 and 2 I'm going to put with the protein there. Okay, so we've got glucose and Benedict's, we've got starch and iodine, and we've got protein with Biuret 1 and 2. The last one we've got is lipid. Okay, so I've got an empty test tube for this one, and I've got my known sample of oil, so fats. Okay, just gonna put a little bit into here. And what we need to do for this one is we need to add a small amount of ethanol for this test. Okay, so just with a pipette, I'm going to add a small amount of ethanol. Now, if you remember from the kiwi experiment, we used ethanol as well. And what it does is it separates the layers, if you remember. It does exactly the same here. This is called the emulsion test. And basically, it should separate into a milky kind of layer. You can see the two layers there. Give it a couple more. Okay, so I'm now just going to add a teeny bit of water. You can already see the emulsion. Okay, I'm just going to add a tiny bit of water. And even better, you can see the emulsion forming there where the oil and water don't mix. So the white colour means that a lipid is present. Okay, so the oil and the ethanol are together. So that's your fourth test. So if we have a look at all of your tubes, we've got the Benedict's blue going to the copper, the orange brown colour. We've got the iodine going from typical orange iodine to blue black. We've got the beautiful lilac of the Biuret test for protein, and then we've got the emulsion test using ethanol for lipids at the end. So that's your 4 food groups: glucose, starch, protein, and lipid or fats. Those are the 4 practicals you need to be able to describe and explain how they work for this section.